disease-resistant traits and genes, as well as abiotic stress tolerance traits. This study seeks to close the knowledge gap where the Mercury Hill rice genome has not been sequenced before, nor its genetic potential has been untouched to this day. And no publications are out there that identifies the ancestral origins of this Mercury Hill rice. And in terms of the significance of the study, it lays the firm foundation upon which we could do more research and develop new elite varieties that are climate resistant, has a broad range of disease-resistant traits, all while being abiotic stress tolerant, as well as low input for the farmers. So we want to develop something that we can give the farmers that we could say, this is ours, it belongs to Trinidad, and not rely on imported rice from Guyana. I know Mr. Fortuny, he mentioned that we need to be self-sufficient and produce our own food. So this is a foundational study towards that. So we wish to unlock the genetic potential of the Mercury Hill rice, and firstly, we have to assemble and produce a highly contiguous genome that will structurally characterize and functionally annotate the genes. We will also perform gene mining and for putative disease-resistant genes and stress-tolerant traits. And then we will go on to identify the closest African origins for the Mercury Hill rice. So we grow the rice to the F3 generation, and we extracted genomic DNA from the leaves of this F3 generation. We used the unmodified CTAR protocol, and we went on to perform two types of sequencing. Long-read sequencing on the Oxford Nanopore Gridiron platform, and this was done right here at the University of the West Indies. And we did short-read sequencing on the Illumina NovaSeq platform. This was outsourced to a company. And to make this method a little bit simplified, I want to draw the analogy of using a picture puzzle. Imagine this picture puzzle it's special that it has large pieces and small pieces. So it is analogous to the long reads of this genome as well as short reads. Firstly, we put together all of the long reads together to form the bigger picture, and then use the short reads to fill in all the different gaps, and then use those short reads again, which are high quality, to polish the genome to produce a highly contiguous assembly. These contigs were then scaffolded to the reference genome for Oryza glabrema, ORGLA-RS2, and then we use this assembly to annotate genes, transposable elements, and screen for NLR genes, which are indicative of disease-resistant genes in the genome. We perform gene ontology analysis and genomic variant analysis, where we use those variants that, along with the variants from a panel of 112 different African land races, to identify its ancestral origins. So when we performed short-read sequencing, we got 137 times genome coverage. And for Oxford Nanopore technology, we got 33 times genome coverage. On average, these coverage values were on par with other studies, but actually in the case of the short-read sequencing and Illumina, it was much higher than other studies we found. This is a summary of the assembly statistics, and we performed BOSCO analysis. BOSCO analysis is an evaluation of the completeness of the genome. The Mercury Hill rice was 97.4% complete. And for comparison to the reference, which is what we consider as the gold standard for this African rice species, it was 99.1%. So just a small value off. This here is the first Mercury Hill rice genome assembly that we assembled together. And we found that it has a genome size of 379 megabase pairs. And we found this to be about 18% larger than the reference genome assembly, but it only just had 1.79% genome gap. This is very small compared to other studies. And we found just over 56,000 genes in the genome, and while this was much greater than the reference assembly, we found it to be more comparable to other studies on African rice cultivars. This is a summary of the genomic repeat elements in genome, and we found that just over half of the Murray-Hill rice genome was littered with repeat elements. This is not unusual because in general, plant genome has a lot of repeat elements, and some of them are responsible for gene regulation, especially for disease responses and abiotic stress responses. We performed genetic variant analysis, and we found just over 3.5 million variants, and of this, 2.9 were single nucleotide polymorphisms, or SNPs, while the others were insertions or deletion sequences. And we found that the SNP density was much higher than other studies. And this would become more important when I look at the ancestral origins of the Murray-Hill rice. Just over half of the variants occurring in genes were non-synonymous and missense. So in simplified terms, these variants causes the proteins and transcription factors to have alternate forms. Gene ontology analysis showed that the Murray-Hill rice does indeed has a lot of genes associated with biotic and abiotic stress tolerance traits. So nucleotide binding leucine-rich repeat genes, these are genes that are indicative of disease resistance in plants, and the Murray-Hill rice was found to harbor 474 NNR genes. And of these 474, 9 of them were found to be homologous, or almost exactly like PI genes that are responsible for blast resistance in rice. And one of them was found to be homologous to a gene that confers resistance to sheep blight disease. When we did the analysis to check for if any of these NNR genes were also homologous to blight resistance, bacterial blight, we interestingly didn't find any natural relations. So we can summarize this to say that the Murray-Hill rice has a lot of potential for blast disease resistance. We found several abiotic stress tolerance genes, heat shock proteins, peroxidases. All of these have been reported in literature to confer abiotic stress tolerance traits. And we found that the origins of the Murray-Hill rice was most closely related to a Nigerian accession. This accession was IRGC 10494. But despite this close relation, you can see from the PCA plot that it still remains distinct, and we can attribute this because it was grown in Trinidad for over 200 years. And because of this geographic isolation and environmental factors, it became its own. So in conclusion, we found that the genome size was 379 megabase pairs with just over 56,000 genes, and the Murray-Hill rice harbors a lot of disease resistance and stress tolerant genes. And we found that it was most closely related to the Nigerian